Nothing gets people in the seats like pictures of Petri plates, right? So, okay, so I'm Tyler Bourret. I'm with the Rizzo lab at UC Davis Department of Plant Pathology. And when I started my PhD 5 years ago, it was sudden oak death was the main disease I was working on. And I since still have some projects with that. But I did a whole project sponsored by Santa Clara Valley Water District here on sites with Phytosphere sort of following up with their work and teaming up with them on this restoration project. And it's been the most satisfying science that I've ever done in my, it's getting longer career. I'm really close to the end of my PhD. I'm going to turn in my dissertation before September. And one of the chapters, yeah, don't applaud me yet, but thank you, yeah. So anyways, I want to talk about, I'm at the end of my PhD. My job is identifying Phytophthora to the species. That's a common thread of every one of my dissertation chapters. And so I want to talk about where we're at. I promise not all of my slides will have this much information, but I brought my prop here. This is the Phytophthora Bible, Phytophthora Diseases Worldwide. And we're lucky to have this. You know, plant pathology is really an ad hoc scientist. The reason we started studying Phytophthora was because of the Irish potato famine. And the reason I got to do my PhD here is because of sudden oak death and the desire to manage Phytophthora species by species and know that not all Phytophthora species are created equal. And this is really where we start. And so I just wanted to highlight some writing from, this is what the Holy Bible says about Phytophthora cactorum. It has a wide host range, we know that. And I think this is a really interesting point that on this page that are really a subtle point, but it really gets to the heart of the scariness of this problem, which is Phytophthora cactorum strains, they have a very wide host range, but they don't necessarily specialize on one thing or another. It's really hard to predict. And so that allows them to proliferate in nurseries because they can move from one host to another. But it also means that if they escape into an ecosystem that's naive to that particular species, there's always this chance that the interaction they have with one of those native plants is going to be just really strong and really virulent. And, you know, that's really what sudden oak death is. And so this is what we have to watch out for. One thing, just because I think it's really neat, I believe that this last piece of work that's mentioned, that forest soil isolates of P. cactorum don't infect ginseng, that's Jennifer Park, right? That's earlier work. So you'll get to hear about me about all of these deficiencies, and then you're going to get to hear about Jennifer about actually building up the knowledge and real stuff on the ground. So this is from 1996. DNA, the analysis of DNA sequences has really come into play since then, and we've learned incredible amounts about Phytophthora, yet so much of what we used to know is sort of more fragile, or really we know we kind of understand less than we did. And the big picture of that is because we are moving from a system in the way we identify and classify Phytophthora species from looking at them under a microscope and doing the best we could do under that to using DNA. The result of that is the amount that we thought that we knew has declined. And so one of the big reasons is because we used to think that we kind of had a idea of the general Phytophthora diversity. For most of the 20th century, it was kind of in the 50 species or so. In this book, there's about 65 species. Since 2000, we've described another 150, and yet at the same time, we think we have found less of what's out there. So we think that there's probably 600 to 1,000 species out there, and at most we found a third of them. And so that's a big reason that we think we know less than before. So getting a little bit more into why that might be, one of the reasons we've found so many more species is just going out to natural ecosystems and looking. And that's something that didn't happen as widely until some of these diseases like sudden oak death. But one of the other reasons is this concept of cryptic species. So we had definitions of species based on how things look. Now that we have other tools, we can see that actually what looked to us like one species is actually multiple things. And if we go backwards in time, they all would have been identified as the old species. So here, and this is gonna be my main example. We have all these things that look like Phytophthora citricola, but we can see they are varying levels of different than Phytophthora citricola. And so that means that it's very hard to go back and know what we already knew. Go back and look at the old literature about distribution, pathogenicity, and really interpret it with what we now know. So that's a big problem. Why are we doing this painful transition? And besides breaking this compatibility, it requires more expensive techniques. It requires some amount of additional lab capability that not every lab might have. So the real reason we're doing it is because it is a tool unlike any other that biologists have ever had, and it is more precise. So what we can do with it is better than we ever had before. We can see those cryptic species, and we can really begin the thing that we really need to do this. People have already sort of asked for this. We need a comprehensive list of native, resident Phytophthora species and exotic Phytophthora species. We have no hope of early detection and eradication if we just have no idea where these things are from. And yet, on the other side, if we can't ever pin down which things are resident, if we can't ever pin down which things are resident, then the people who in charge of regulation are just going to have to say everything is a potential terrible risk, and we're going to have to. We're not going to have any, consistency about how we deal with each species. So DNA is a great tool. Someday it will also give us something that it is lacking, which is consistency of species lists between researchers. And I think this is a place where we're really struggling, and I'm going to go into more about why that is. But before I get into that, I want to talk about a little bit going into the future, although the future is now. And the real hidden benefit of this growing pain of switching over to using DNA is this tool that goes by various names, meta-barcoding, culture-independent techniques, where instead of isolating Phytophthora, we've already heard that one type of bait, pear, rhododendron, isn't enough to find every species of Phytophthora. And so forget it. Let's just look for them by the signature of their DNA. And one example from the project that I worked on, the Rizzo lab with Phytosphere and SCVWD, was this really important species, Phytophthora quercina. We found it for the first time in North America in our project. It was #1 on a list of species we hoped to keep out of the continent, or out of the country. And the problem with Phytophthora quercina is it's one of these species that doesn't take a pear bait or a rhododendron leaf bait. We kind of were serendipitous in that we seemed to be the first and only time it's ever taken a pear bait. Once we realized it was there, my colleague Heather Mell read, it only likes oak leaves. We went out, we baited with oak leaves. We were able to establish it was on several valley oaks at this one particular site. But really, the overall context of this species is unknown, and it could kind of be there. We're not going to find it until we go looking for it. So to get a little bit farther into the nitty-gritty about some of the issues here, the sort of main reason that species lists are not consistent researcher to researcher is species boundaries. There are some Phytophthora species where we only have one exact sequence. You match that sequence, it's always the same, very easy, it's always that species. But many times, one of the problems that people like myself or Suzanne run into is we get a sequence from a particular strain. That sequence is new, and we can know this because we have this database of all published sequences and some unpublished called GenBank. And it's pretty close to species A, it's a little farther away from species B, but it's distinct from one of those. So what do? Do we call it species A and expand the definition of species B? or species A? Do we actually expand species B? Or do we decide we need a new category to accommodate this new thing? And there is no, even with the old system, there wasn't necessarily an agreed upon way of dealing with these ambiguities, but certainly the DNA system is so new, there isn't a school of how to use DNA sequences to identify any type of fungus or oomycete. And so we just don't know. And so different researchers, you give them the same set of strains, they are very likely for doing things the right way. Nobody's right or nobody's wrong. They're going to get different species because it's a bit of an art, if you will, this identification right now. And I'm like, oh, what are you talking about? This is supposed to be science. And we know, even those of us who do consider ourselves identification artists, we would like to automate this. And there are some tools that are beginning to do this, and where we can get some consistency. PhytophthораID.org, PhytophthораDB. But it's still a little bit difficult. Species are being described so quickly that even if you use the same database, if you look at your results, you look at the same results a year later, you might get quite different results from the exact same tool. So you can understand how this is difficult. So going down even farther into the specifics, and I promise we're going to come up for a breath of air in a second, talk about an example. But what are species? We care so much about them. It's the unit of regulation, which becomes very important typically. So we do really have to know them. Where do they come from? Well, they come from Linnaeus, and they come from the 18th century, and they predate evolution. So when species were invented, we didn't think of them as evolutionary units. They were just the smallest box you could put something into. And, Linnaeus had the animal kingdom and the vegetable kingdom and the mineral kingdom. He was classifying rocks right alongside living organisms. And so even after Darwin came in more than 100 years later and gave us this idea of common descent and everything was related to each other, it still took a really long time to understand how species fit into this story. And the story is really cool. It's part of this amazing modern synthesis of biology. And species are so cool because they're one place where ecology and evolution interact, right? So the species evolve and they interact with the environment through the niche, this idea of niche, and it's just really cool. Ernst Mayr, a zoologist, as part of this modern synthesis, came through with this great concept that is so elegant. It works great for animals, the biological species concept. And so part of this idea is that species are real, they're evolutionary entities, and if we can figure out how to define them, then we can go out and measure them, and we can actually have an explicit criterion for dividing up species and establishing these boundaries. And so the classic example is the mule, which proves that the donkey and the horse are different species. So donkey and the horse can mate, they can produce an offspring, but that offspring is sterile. And so that proves that they're reproductively isolated, and under this context, under this criterion, that means they're separate species. So that's great. It works great for animals. Imagine, though, if that mule could asexually reproduce. And so, yeah, it can't go back and mate with the horse or the donkey, but it's still around. You see it. It's out there in nature. You may encounter it. You have to give it a name. And let's imagine that, even more complicated, the horse and the donkey, they can mate with each other, and they can produce a whole host of different hybrid offspring, what we would call a hybrid swarm. And this is the reality. And in fact, this idea of more complex species, it comes from botanists because, sorry, thank you. Because plants, we look at a genus like Quercus, the oak genus, this is a common thing. It hybridizes, the offspring are fertile, yet at the same time, we go out in nature, we can see these species are distinct entities. And so we had to come up with a more complicated criterion or different sets of criteria that can accommodate this more complicated species concept. So bringing us to Phytophthora. Phytophthorologists are scientists. We have to we do believe that Phytophthora species are real, just like a good botanist or zoologist. But we're still learning how to effectively measure these real entities. And so it's limited to some of our more important species. So just a quick history of Phytophthora in this context. When we first started knowing Phytophthora, we didn't have a good idea of how the species concepts work. And so we didn't even really know if microbes had species. This sort of microbes were still new on the scene. So one thing that made a lot of sense to a lot of researchers, Phytophthora is a plant pathogen. It's probably host specific. A lot of plant pathogens are. So you find a new Phytophthora on a new host. I got a cactus. Hey, it's Phytophthora cactorum. And so there was this early period of lots and lots of Phytophthora species. Every new Phytophthora host had a new species. Unfortunately, that didn't work for everything because a lot of the species have very wide host ranges. So there was a move to get all these different species, get them in a pile, look at them, and say, no, all of these are the same. And so we moved to our morphological species concept. And this is really what persisted and what got us to where so much of our knowledge of Phytophthora is based in this period on understanding species that are defined morphologically. And then finally, bring us to the present, we have all these new tools, but we still haven't figured out exactly how to work them. What we really need is a DNA to species translator, and we are working on it. These are our closest bets, but they're still not, they're still in process of being developed. So we don't have a good way of biologically measuring species because Phytophthora is complicated. It likes to have sex with itself or not have sex. Even species that are outcrossing, they'll typically mate with any other species of Phytophthora. And so it becomes very difficult, sort of its own study unto itself, to separate every pair of Phytophthora species and every complex group of closely related species. So bringing us to and I'm nearly done with this story, and we can talk about the real examples. What are we doing right now? So we have this new tool. We don't want to give up the new tool. It gives us all this precision. So we have back to an artificial species concept. Essentially, if you can show based on sequences that what you have is different than anything that's already out there, then you're cool calling it something new, describing it as a new species. That's the criterion. And that's great. It gives us the opportunity for all of this diversity that we're able to see to get it out there into the literature, which is really important so that we can all be talking about it. But it still doesn't give us a way of grouping things. It sort of forces us to put things into the smallest categories that we can discern. Nothing wrong with that necessarily. Precision is not bad, but it's one of the reasons we have so many new species that are coming out, because each one of these little entities are being described as new species. That puts scientists in a bit of a bind. We have to go out and publish in scientific journals, and the people who are moderating those journals, they're scientists, they are living in this world of real species. So if we come through and say, well, we've got all these new species, but they're defined in this way that we know to be artificial, that causes problems we might not be able to publish them, which is the only way we can really get them out there for everybody to know about. So phytophthorologists have come up with this sort of provisional system. So we don't know if these species are real for various reasons, but we want everyone to know about them. So we've given these sort of placeholder names, which is certainly annoying if your job is to make lots of lists, because it gives things multiple names, but at the same time, it allows us to fudge not going out and actually measuring these real species and delivering all this new diversity that we're seeing, so we can start talking about it, both scientist to scientist and scientist to other folks. So that's all the background. Hopefully that is interesting information, relevant information for making sense of all this. I wish I could have be sitting in this audience 5 years ago at the start of my PhD. So let's talk about an example that I think really is pretty straightforward, and it really shows how we're dealing with all these issues. So UC Davis campus, back in the 70s, we have all these great oak trees, including the cork oaks that were planted during World War II. and something I learned about from Linda Spar, who's a SCVW employee. I don't know if she's, anyways, pardon me. So these great phytophthorologists, my predecessors at Davis, studied the Phytophthora incidence on two different oaks on campus, cork oak and coast live oak, or as we like to call it in the Rizzo lab, quag. And so this is one of their figures from this great picture. They found Phytophthora cinnamomi on the cork oak, and they found two species, Cactorum and Phytophthora citricola, on the coast live oaks. And just to bring you up to date, they recommended the grounds crew. You can see that back in the day, the lawns went right up to the bases of the trees, and they were using sprinklers. So they pulled back the lawns, they put in decomposed granite, stopped letting the sprinklers hit the trees, and most of these trees are still alive and still iconic parts of the campus. So as part of our practicing our baiting techniques in the Rizzo lab, we have a particular oak, a coast live oak in front of Herring Hall that we have periodically baited from to see what's there. And in my time in the lab, we've done that 3 times, and we've used DNA sequences to identify what we found. And so we found these 4 species. The Phytophthora multivora we found in every single time we've looked at this coast live oak. To make the story a little more interesting, last summer, this coast live oak, a different tree that's also really close to us in front of Storer Hall, started declining. It had bleeding cankers, and by the midsummer, it was totally moribund, and the grounds crew took it out, cut it down, and it kind of, Storer doesn't look the same anymore right now. They didn't put anything in there. Last fall, we baited Phytophthora acerina from underneath the stump of this tree. And then this January, during a windstorm, the one we've been baiting from for quite a few years blew over, and my boss, who Dave Rizzo, took a look at it. He said it was, I forgot to ask him which actual fungus, but it was a butt rot. A true fungus was really the thing that caused this to blow over. But we know it had Phytophthora underneath it for many years. It's next to a planting. It gets a lot of water. So we had these dead trees. We had a list of Phytophthora species. We have this old study. That's really cool. So can we reconcile the old study from the 70s with the results that we have now, given that we use 2 different methods for identifying the species? So here are our lists. We got Cactorum in both times. That's great. That makes sense. What about citricola? What happened to Phytophthora citricola? How about where did we get these other 3 species that we found? And this is where all of this issue with the cryptic species comes into play. So if we look at an excerpt of the phylogeny of Phytophthora, and we put species that look like Phytophthora citricola based on their morphology, in orange you can see there's quite a lot. And some are closely related to the true citricola, but other ones are actually pretty distantly related, yet they still looked the same. So putting our 3 species that we found in 2010 onto that, they all could have been Phytophthora citricola from that paper in the 1970s, but of course, it could have been none of them, it could have been one of them, it could have been something that's not on this list because we have yet to find it, at least in the molecular era, and get a sequence from it. So we have nearly a century of research about Phytophthora citricola, and it's almost impossible to properly interpret it. The only real way to do it in this current context is if the people who did those studies in the past saved their strains of Phytophthora, and they have subsequently been sequenced, and we can put one of these modern names on them. Without it, we really are in a position of not knowing very much at all. And we checked. We're probably a couple decades too old to find the strains from the 70s campus paper. We looked through Greg Brown's collection, but they didn't exist anymore. And so imagine you are reading an old study and it says Phytophthora citricola is native to California. It's resident in California. Well, what does that really mean? Which one of these species are they talking about? If someone gives you a shrub and they said this shrub is resistant to Phytophthora citricola, which, if any, of these species is it really resistant to? this is really the heart of what we're dealing with right now, why it's very difficult for me to even say any species is native to California. That actually becomes a very difficult question for us to ask. And these new tools we're getting there, but there's this growing pains that we're going through. So I want to leave with another case study, thank you, that is a little bit more hopeful. So the story with Phytophthora citricola seems a little scary. One of the reasons is because for about 100 years, we thought we knew what Phytophthora citricola was. Well, here's an example of two species that, for the life of these species, phytophthorologists have struggled to really properly discern them and identify them. These two species, Phytophthora drechsleri and Phytophthora-cryptogea, they're part of what we call one of these species complexes. When people study them, you could always find, two strains that looked really different. And you're like, okay, there's got to be more than one species here. But then when you study them a little bit more, you find things that are, in the gradation between them. As people use different methods, they thought they could divide it up better, or they could collapse them into one species, and then someone else would come out with a new method and undercut those results. And we added a few other species to this group, including the one Phytophthora erythroseptica that Ted was mentioning earlier. Anyways, but even when the molecular era dawned in 2000, it didn't really lend any sort of sanity to this group. And when I started my PhD, if we encountered members of this group, I would just say, oh, they're in the drechsleri-cryptogea species complex. And I can't get any more specific to that. And if anyone gave you a more specific determination, they don't know what they're talking about. So that's where we were. And of course, there are still plenty of species complexes where that is exactly the case. But in 2015, this excellent study came out that really replaced nearly 100 years of ambiguity with a lot of clarity. And all of a sudden, things now, we can really sort them out, and we can start to make sense of a group that has really been fuzzy for its entire existence. Group took the time to assemble a worldwide collection of relevant strains. That's really important, and it's really difficult because these are quarantine pathogens. For good reason, we don't want to just be sending them willy-nilly across continents without a lot of care being taken. And they essentially, they used a lot of different genes, and they also used other methods. So we like to use the morphology too. And they were able to find 5 different species out of this group, and they needed to add this Phytophthora pseudo-cryptogea in order to accommodate a new group. But these are nicely well divided, and I can identify them with the sequences that we use. And so all of a sudden now we can take the results that we have in the Rizzo lab and start asking questions that we couldn't ask before about the natural history and the distribution of these species. So we have an ongoing stream monitoring project to look for the movement of the sudden oak death pathogen, and we have a lot of diversity from that project, including members of this complex. But when I looked at it, all of the strains were this new species, Phytophthora pseudo-cryptogea. Likewise, the project we did here with SCVWD, mostly pseudo-cryptogea, and then this one provisional species, Phytophthora species Kelmania, we also got one time. The 3 oldest members of the complex, we have not encountered them. Now, I'm not going to say that they don't exist in California. This is just my lab, and we're just beginning to start this. But you can see how one species complex at a time, when the work is done, we can actually really, perhaps Cryptogea, let's just imagine the true Cryptogea, sensu strictu, is never been found in California before. Even though this is seen as a ubiquitous, widespread species, if it's not in California, we need to be monitoring it. And so this is what we are beginning to be able to do. One more example, just because it's relevant to this story, and then I'm done, I promise. Phytophthora megasperma is another species that was described based on distinctive morphology. The megasperma means big seed. It has the oospores, which are the sexual spores of Phytophthora. It has huge oospores. One thing that was really noticeable to the early phytophthorologists about megasperma was, unlike Phytophthora cactorum, where you have any particular isolate you have will likely have a wide host range and a sort of unpredictable one. These megasperma isolates, if you isolated one from soybean, it tended to be mostly just pathogenic on soybean. And if you put it onto a Doug fir, for example, it wasn't that aggressive. Yet the ones you got from Doug fir, they were aggressive to Doug fir, not so much to soybean. So there were host-specific groups, but we couldn't divide them up based on our DNA abilities. So over the course of the 20th century, using all of the fanciest tools that anyone could bring to bear, 3 of these legume-infecting species were pulled out of the megasperma complex, if you will. And one of them, Phytophthora soji, is one of the most economically important Phytophthora species for agriculture, for soybean production. More recently, some species that have really become important, they really were figured prominently in the survey that we conducted here in this area of restoration outplantings. Phytophthora rosacearum was a group that had been seen as specializing on fruit trees. So a lot of, stone fruits are in genus Prunus are in the rose family. And that was separated by Everett Hansen's group at Oregon State, along with another species that, weirdly enough, infected conifers and soybeans. And then that brings us to more recent times, this Phytophthora crassimira, which was described in the course of the survey we conducted, and turned out to be, besides Phytophthora cactorum, the second most widespread common species in our survey. And it really is surprising everybody. It's a member of this clade 6 group that, to a large extent, we don't see as being particularly pathogenic. But man, it's devastating natural ecosystems in Europe, and clearly a big part of this restoration story here in California. And interestingly enough, if you see where all these Phytophthora species that looked like megasperma landed, this is the whole genus. They're kind of all over the place. This is the the legume, 3 of the legume species. Oh my gosh. That clearly means it's time for me to wrap up. Anyways, crassimira is the only one that still is very closely related to Phytophthora megasperma. Phytophthora megasperma is still a complex, so it still exists, and we have found a member of that species also in this. It still probably will. I'd say there's probably at least 5 species that will emerge from it before we're finished with it. With that note, I want to skip through just talking about the future. Hopefully this is not a doom and gloom talk. Things are moving really rapidly, even though it is like literally my only job not my only job, one of my main jobs, I also TA a lot, to pay attention and be on top of this story, all these new species. It is still very difficult, even for somebody like myself who not only is good at it, I would say, but loves it. The species list we produced for our survey that we did with Janelle, it changed over the course of that survey because so many species were described during the course of the survey. That's crazy, and even a scientist shouldn't have to deal with all of that change. Eventually, this is going to slow down. I don't know when it will be. I would say we probably have at least another 10 years of quite rapid new Phytophthora species, including, I'm going to describe at least 5 new species as planned for my dissertation, and that's just a fraction of what I have in my hands from various labs, and I work with the CDFA as well. We sort of consolidate our strains. But this is this is the road to really understanding native species. And I actually think that one of the species I found is definitely a native Phytophthora species, very closely related to Phytophthora cactorum. We found it from Big Sur forests, so undisturbed natural ecosystems, and that will be, if it is truly native and we see it and we can study a Phytophthora in its native ecosystem, that's essentially going to be one of the first times we'll ever have that opportunity to do that. No, that's, oh, God, really important acknowledgments. I think you guys are familiar with all of these folks that I'm acknowledging. Some of them are already talking, but the CalPhytos portion of this project, for me as a scientist, has just been the reason to get out of bed. It's a reason to stay in the lab really late, and you don't expect that what you do in this dry world of science is ever going to translate to any sort of finds on the ground. But I really feel that I have been part of actually keeping forests safer as a part of my part of this project, which is just no other forest pathologist gets to say that. So, thanks.
The actual only part of my job required for the moderating was to let you guys know that we actually do have 5 minutes available for questions. Questions?
About the megasperma. What was? Can you go back to that slide? Because
Sure.
there was a term you used that I couldn't really wrap my head around. Let's see.
This one I have two. There's.
It was like lumping things into like a smaller group, whatever. Like you had the entire genus and then megasperma, something with other species that were in this megasperma group.
Yeah, so good question. You sort of called me out on a bit of a red herring, but there is a story there. People have wondered, this is a phylogeny of Phytophthora. We have there's about 120 species on this phylogeny, and we can divide Phytophthora up into these, what are called clades, and there's 10 of them, although look to my first chapter of my dissertation for 5 additional ones. But, that's a separate issue. One question, the ability to to sort the Phytophthora species by these clades, if you will, phylogenetically, it's a new tool. And one thing, in a lot of other groups, a lot of fungi, other microbes, being able to sort them out phylogenetically has made a lot of sense. It's like, oh, this clade is the group that infects this host. This clade, that has really not worked out for Phytophthora. People have been trying to find this hint of how phylogeny interacts with the Phytophthora as a pathogen, and it's not a very elegant story. The only really good example where ecology is something that is sort of inherent at a clade level is this clade 6. And clade 6 is a really interesting clade because the majority of diversity we know from it is from sampling aquatic ecosystems. And in Irwin and Ribeiro, there were only 2 species that existed in clade 6. Now there's like 50. So it's also where a lot of our additional knowledge of diversity has come from. And besides Phytophthora megasperma, which was certainly in this book, none of the species were really seen as being very strong pathogens. And I have a colleague in the Rizzo lab who spent his entire PhD, he's also finishing up, Kamiyar Aram, looking at the fact that these clade 6 species are often resident. They eat decaying leaves in aquatic ecosystems. And of course, this is a relevant thing if you're managing riparian restoration sites. This is a real factor, which is that there are resident Phytophtheras. They may still be pathogens. We don't know where they came from, but they're in the water, and every time you have a flooding event, they're going to be moving around. And perhaps they still have the ability to infect plants, so maybe they only infect weakened plants under the right circumstances. They're kind of... so anyways, finding Phytophthora crassimura a clade 6 species, and in fact rosacearum, which was fairly widespread and quite aggressive from our project. It really has been a big part of this survey. Clade 6 species are no longer just seen as, we should no longer consider them to just be saprotrophs, decayers. They definitely, under the right circumstances, can cause disease. Even some of our oldest species, Phytophthora gonopodoides, which is the one that was seen as the old saprotroph, never causing disease, in recent years it's been associated with massive dieback of almond trees and other fruit trees in Europe. So even old knowledge, even these species we said, oh, they're never going to be pathogens, under the right circumstances, the combination of drought and disease, or and a pathogen, you set up this situation where it's really hard to make predictions.
Hi. I work in the coastal forests, and so you piqued my curiosity with the native Phytophthora in big sur forests. And is it causing diseases?
Great question. One of the hardest parts about studying native pathogens is that they are inconspicuous in their native range. So if you go out looking for Phytophthora in coastal forests of California, you will see one species over and over. It's Phytophthora ramorum, and it causes sudden oak death. It's in the middle of an epidemic, it's exotic, and it's everywhere. To find a native species is much more difficult. And so I was not looking for this. I was actually looking for Phytophthora ramorum, and I found this other species. We baited it from bulk soil, so we haven't conducted pathogenicity tests, but it was not associated with any root Phytophthora disease. Some of the plots we got it from are also infested with the sudden oak death pathogen, but that's an above-ground pathogen. We consider this to be probably below ground. But once again, I really don't know that much about it. The reason I think it's native has to do with the DNA sequences, actually. It has a subtle variation of, I have a population of 12 different strains of this new species, and there's a little bit of variation, but a significant amount that suggests it's not clonal. So species we know are non-native in California, this Phytophthora ramorum, some of these other species, their DNA is very uniform because they're the result of an invasion and they haven't had much time to create variation. I call it Phytophthora sequoia soil. If I'm going to describe it as a new species, it'll be Phytophthora sequoiarum of the sequoias, I think. So that's why I think it's native, is through this subtle variation that I see in its sequences. But just to follow up on this, it's very closely related to Phytophthora cactorum. Phytophthora cactorum, if you go to the excellent website, Forest Phytophthora, it's seen as a ubiquitous, widespread around the temperate ecosystems. But if you look a little bit more carefully with these new tools we have, making the statement that Phytophthora cactorum is resident in California, I wouldn't back you up on that. I think that's a really scary thing when you see that how big of a problem it was in this particular story. And just one more wrinkle, Phytophthora cactorum is a bit of still kind of a large species. There is one seemingly clonal strain, and that's the one that is associated with all of these restoration projects. It's the one that seems to be in the restoration nurseries. If you look in what's out there in the databases, we've never seen that before. We don't know where it comes from. So even though it's part of this species that we theoretically know so well, I would be very afraid if it gets out into natural ecosystems. And so this idea, Phytophthora cactorum is resident, no problem, I would throw that right on its head.
Just a quick comment about rosacearum. As you mentioned, it was prevalent in our study and also common on fruit trees. And I find that really interesting because the history of Santa Clara Valley is it's mostly was planted with fruit trees before it started being developed with Silicon Valley. It was called the Valley of Heart's Delight because of all the fruit trees and agriculture. So I'm wondering if that came in on those trees hundreds of years ago. Just an interesting connection possibly there.
Absolutely, yeah, that's a great question, and it's not a species that has been found very widely. It's not widely distributed as we can see from any source of evidence. So I don't know, but it's one of those things I can't ever answer. Sorry.
All right, well.